Hi, my name is Cameron Burke, and welcome to this Kaplan MCAT science review of oxidative phosphorylation. Oxidative phosphorylation is a big process that you're going to hear about in other places when you're studying. But in the meantime, this video is going to focus on ATP synthase and its part of the process, as well as the role chemiosmotic coupling has in producing ATP, and finally, the way ATP synthase can be regulated in the oxidative process. Ideally, by the end of this video, you'll understand the big picture of ATP synthase, how it works, and what role it plays in oxidative phosphorylation. If you achieve your learning goal, you'll be able to answer questions like this one about the effect of changing pH in the mitochondrial intermembrane space. Oxidative phosphorylation is a super complex process that eventually results in the production of ATP and metabolic water, and ATP synthase plays a huge role in that production. ATP synthase is the gatekeeper to producing ATP. It's a protein complex that links the electron transport chain with the final ATP product. The MCAT likes to ask about locations of stuff, so keep in mind that it spans the entire mitochondrial membrane and then sticks out into the matrix. Chemiosmotic coupling is basically a process where a chemical gradient is created, and then the energy from that gradient is used to phosphorylate ADP to turn it into ATP. A portion of ATP synthase spans the cell membrane. This piece is called the F0 portion. F0 does the job of an ion channel, so protons travel along their gradient back into the matrix. This is the gradient that allows chemiosmotic coupling to go forward. Basically, the electron transport chain makes a high concentration of protons in the intermembrane space. Then the protons want to flow through F0 back to the matrix. As this is happening, the F1 portion of ATP synthase uses the energy to phosphorylate ADP into ATP. The proton concentration of the intermembrane space is vital for chemiosmotic coupling, because there's a direct relationship between the energy from the gradient and the phosphorylation of ADP. The protons travel through F0 spontaneously in an exergonic reaction, and that energy is used for the creation of ATP, which is an endergonic process, so a more energetic molecule is the product. While biochemists think chemiosmotic coupling has a direct relationship to affecting ATP production, chemiosmotic coupling could alternatively promote an indirect relationship between the proton gradient and the synthesis of ATP by inducing conformational coupling, which is when ATP is released by the ATP synthase as a result of conformational change caused by the gradient. In this mechanism, the F1 portion of ATP synthase is reminiscent of a turbine spinning within a stationary compartment to facilitate the harnessing of gradient energy for chemical bonding. ATP synthase is where the oxygen we breathe comes into play. If you'll notice in the illustration, as ATP leaves ATP synthase, so does metabolic water. The MCAT likes to test on the role of oxygen in this process. The spare hydrogens created as a byproduct from making ATP need to go somewhere, or acid starts to build up inside your cells, and that would be really bad. So they get stuck on oxygen inside ATP synthase to create metabolic water along with ATP. And for this reason, oxygen is one of two key regulators for this piece of oxidative phosphorylation. Because of its vital role, oxygen is one of the stars of the show in terms of regulating oxidative phosphorylation. There are actually many places where regulation can take place during the entire process of oxidative phosphorylation. But for ATP synthase's role, there are two key regulators, both of which basically hinge on respiratory control. In addition to oxygen, we also need ADP for this process. The first scenario to consider is when oxygen is low or you don't have much ADP. And the second is when both oxygen and ADP are plentiful, so you have as much as you need of your key ingredients. When you're short on oxygen, oxidative phosphorylation decreases, and the electron carriers from the citric acid cycle start to pile up because oxygen's job is to catch all those hydrogens eventually. So if there isn't enough oxygen, the hydrogens have to stay put on their carriers in the matrix. The backup in the matrix inhibits the citric acid cycle to stop producing so many proton carriers until oxygen levels are back up. When you have enough oxygen, ADP is the main force affecting the regulation of oxidative phosphorylation. A lot of ADP buildup means you're short on ATP, because ADP is just ATP minus a phosphate. So when ADP concentrations get high, that means you're low on ATP. The ADP buildup activates the citric acid cycle, and more electron carriers are produced for the electron transport chain so oxidative phosphorylation can proceed. And then oxidative phosphorylation increases. Now that you've gotten a review of oxidative phosphorylation, check out your progress with the learning goals with this practice question about pH changes. The correct answer is D. If the pH in the mitochondrial membrane space increases, the gradient is diminished, so ATP synthesis slows down. Recall that when pH is high, it means that hydrogen ion concentration is actually low. The MCAT loves to test on that concept, so keep that in mind on test day. The second practice question tests your knowledge of respiratory control. B is the right answer for this one. If NADH builds up, the citric acid cycle stops, so there isn't any more ADH being produced until the backlog is cleared. To recap, ATP synthase is the workhorse for making ATP. F0 acts like an ion channel, and F1 is the machinery where ADP gets converted to ATP. Protons create the gradient that allows for chemiosmotic coupling and for conformational changes to ATP synthase. Respiration is the main force for controlling oxidative phosphorylation. When O2 is down, oxidative phosphorylation decreases, causing proton carriers to accumulate and inhibit the citric acid cycle. When ADP is high, the citric acid cycle turns back on, proton carriers are produced, and oxidative phosphorylation increases. Thanks so much for watching this review of oxidative phosphorylation, and we hope it helps you out if you get any questions like the ones you saw today on the MCAT.